bind on T cells. If you're a helper T cell, you have CD4. If you're a cytotoxic T cell, you have CD8. There's patterns when we look at these receptors. Families of receptors often have the same chemical structure, backbone, that hold them together. You'll notice here in all these little bubbles that says C, that's referring to cysteine, right? And cysteine is an amino acid that has a sulfur group as its R chain, using that for bonding, co-bonding to other things. So that dictates some of the chemical properties that we have here. But they all belong to this family because they all have relatively the same structural components and shapes, which dictates their behavior, right? How they work, what they do. This shows you that in the binding portions of these molecules, you have these sort of beta sheet, beta strand arrangements that allow them to interact, to interface with antigens and other molecules to bind onto them. Okay, so here's our antibody as it's released out of the membrane, still exactly the same. The difference here is we have free carboxy termini, which give us a charge, which allow it to be soluble. If the B cell is going to make this as a soluble protein, it, during translation, translates little hydrophobic stalks that then get anchored into the membrane. We're going to talk about that more as we continue to go. But you have this inner anchor chain. You have this outer antigen binds in the pocket here. So B cells can bind two different antigens at the same time, or they can bind the same antigen using both of their arms. T cells can only bind one thing at a time. So right here, this thing that says FAB stands for the fragment that is antigen binding. So that's the hands. Antigens bind into these little pockets. This stalk, this says FC, this is the fragment constant. It's the constant region. It's the piece, the tail piece that holds antibody into a membrane, or the tail piece that can be bound by cells or complement bearing proteins that can bind onto this region. Okay, so this region is just for recognition. This is for binding antigens. The structure itself is there's a mirror image, right? If I chop it down the middle, it's a mirror image of both sides. So it's essentially like a homodimer. The T cell receptor, in contrast to that, is actually a heterodimer. So it's the T cell receptor looks like this. So one side is different from the other side, versus BCR is like two TCRs put together. We'll keep thinking about the structure as we keep talking about how it works. But it's held together by sulfur bonds. There are 5 different types. These are called isotypes of antibody that can be made. I like to think about them as flavors. Right? Ice cream. There's lots of different types of ice cream flavors. They're all ice cream. They're all made, milk, cream, sugar, right? That's the base. But you're going to add a little cream, I'm going to add mint leaves, or I'm going to add chocolate chips, right? To make it slightly different. Same thing, same base, slightly different properties. Most antibody that we think about, typical antibody that we think about in circulation in our blood, looks like this. IgG is the dominant type of antibody. It's moving through our bloodstream, our plasma at any given time. IgA is the kind that is secreted by our mucosa. So the B cells hanging out in the malt, in the gall, our respiratory tract, they're secreting this kind, which is a dimer. It's 2 antibodies hooked up together by their feet. There are a special type of innate B cell that rows around in our peritoneal and our thoracic cavity that only secretes antibody like this. It's a pentamer, right? So it can bind 10 antigens at a given time. This is actually the first kind of antibody that all B cells make before they get triggered for clonal expansion and selection. But they all have slightly different jobs. IgE, this is the one that modulates allergy most of the time. Okay, so I'm allergic to pollen. I make IgE antibodies. IgE binds on pollen. Whenever mast cells, eosinophils, basophils bind onto IgE, they degranulate. That releases histamine. That makes me slimy. IgD. The only one that I'm not going to explain to you long-core about or hardcore about its particular job is because we don't know what its job is still. We do know that a B cell is not mature. It's not ready to do its work until it expresses IgM and IgD at the same time on its surface. So it's more for us a marker of maturity. It tells us, is this a baby B cell or is it a grown-up B cell? It's ready to do work. So they can all have slightly different structures. Most of them are monomers, dimer, pentamer. B cells can make all of these. They're going to be stimulated by antigens, by T cell help. That's going to tell them which type to secrete. This tells me that I name these antibodies based on the type, the structure of heavy chain that they make. So I'm going to back up for a second and just show you that the inner core right here, this blue piece, see how you see CH, 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 VH? This is the heavy chain. It is the core, it is the anchor of the molecule. So based on what set of genes you use to build this core, this anchor, the heavy chain, we then know whether to call you IgD or IgE. What genes do you use to build the heavy chain, the core, the anchor? It also tells us that to make life even more complicated, in the IgG family, there's a whole bunch of different subtypes of IgG that you can make. So isotypes of isotypes, flavors within flavors. Right, like Neapolitan ice cream. You get all 3 flavors in one. It's like that. The light chain. The light chain is this thing, a little accessory helper guy. There are actually 2 different sets of genes that exist in your chromosomes that can dictate how this thing gets built. Those families are either called kappa or lambda. We're going to learn all about this for the next test. Right? But it's just saying, I can make antibodies with different molecular structures. The molecular structure dictates what flavor of antibody I name you. Okay, this is just talking about structurally, right? Variable C. Down here, this stands for constant regions. These are made using patterns of regions of genes that don't change. So I'm going to plug the information from the gene in, I'm going to make the protein the same every time, constant regions. These ends, there's a constant region for the light chain, there's a constant region for the heavy chain. The B's, you stand for variable. It means we're going to mix and match. We're going to mix and match pieces and parts of proteins to make the variable regions. This is going to dictate what antigens I can bind. Right? So constant regions don't change. They act as anchors. Variable regions do change. Those are what are going to actually contact antigens. Right, so that's back here. There is what is called the hinge region. That's just the middle. It holds the arms onto the Y. It can be longer or shorter depending upon whether I'm making a dimer or a monomer or a pentamer. That's it. Depending upon how long that region is, dictates how flexible I am, how wide, how spread apart the arms are. Again, molecular pictures showing like, right, this is exactly how it works. You got a bunch of beta sheets in between those variable regions. It makes a little pocket, it makes a little cradle. And the little cradle is what binds the antigens. When antigens bind in the little cradle, signal transduction gets turned on. This shows you the difference between soluble antibody and a B-cell receptor, one anchored in a membrane. We're going to make a primary transcript, right? So I'm going to take DNA, I'm going to copy the gene regions that I want, I'm going to make RNA. The RNA comes out and it has a little region here that is hydrophilic. It also has another little region beyond the hydrophilic region that is hydrophobic. And beyond that hydrophobic region, we have our poly-A tail for translation. Alternative splicing is what dictates whether or not you make soluble antibody, which cleaves off the green part in the poly-A here, or membrane bound, which includes the green part and poly-A. If I have the hydrophobic region, that gets translated directly into a membrane by rough ER. If I have the soluble end, I can just be translated by a ribosome free-floating in the cytoplasm. Make a soluble antibody that can then be secreted into the environment. When B cells are first made, all they're going to make is this. Then something happens. New genes get turned on. New gene products get turned on. More signal transduction that keys into the cell and says, oh, I don't need to do this anymore. I know what I found. Now I need to pump this out so that it can go bind free-floating antigen someplace else where I'm not. Hormones tell the B cell to do that. Help regulate transcription and splicing. Here is signal transduction at the BCR. I've got my B-cell receptor, my accessory tails that are going to be responsible for recruiting my signal transduction docks, BLNK. I've got a co-receptor that's also being involved in this. I got CD21, which binds complement. I got CD19, which also binds onto a number of different accessory proteins, blah, 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 right? All the signal transduction pathways that we've mentioned. There's RAS, there's MAP kinase, there's PIV kinase, there's NF-kappa B, they're all here. Right? So signal transduction through the BCR can make all of these things happen. Two, depends on what is the antigen, who are the co-receptors, how much is binding, what are the hormones in the environment. T-cell receptors look more or less the same, all right? But we're going to rewind for a second. TCR is just this. It still has constant regions and variable regions. It still has a constant region that's anchored into the membrane, but it's just this portion. So it's a heterodimer, because if I cut this in half, one side is different from the other side. This is a homodimer, because if I cut this in half, the two sides are mirror images. Right? So the BCR is a homodimer, TCR is a heterodimer. I'm going to show you the ribbon model here of my TCR. I got a constant region and a variable region. A constant region and a variable region. And you can't see, they haven't made my ribbons a little bit different for you to be able to see. But there are two major types of the T-cell receptor that can be made. Differentiate by what are the constant and variable regions that get paired up. We're going to learn a lot more about this next test. But still made of variable and constant regions. There's a binding pocket between the variable regions that allow antigen to bind. Here's a better picture. Constant regions. Variable regions. This This is made from one protein piece, this is made from another protein piece. If I cut them in half, they are not mirror images of each other. Okay, so more simplified, just the hands of the B cell receptor. These are all accessory molecules. They don't actually bind antigen. They act as the anchor for signal transduction. So they don't bind stuff extracellularly. What happens is, when this gets activated, it changes its conformation, which makes these guys get phosphorylated and act as the anchor for signal transduction. So here's sort of we're standing looking at the T cell receptor, looking down from the top. Here's the TCR where antigen will plug into. But these little accessory guys are all around the edges, holding on to the base. Right, so TCRs are heterodimers. They can be made using essentially 4 different sets of genes. We'll talk about that later. Members of the immunoglobulin superfamily. Accessories. TCR is always paired with one of these, either a CD4 if you're a helper cell, or a CD8 if you're a cytotoxic cell. Promise, TCR, BCR, all of this will make a lot more sense as we keep moving. But these basic structures bind onto ligands, activate signal transduction, right? All of those pathways that we're talking about. When we come back on Thursday, we're going to talk about the cytokine families. For your exam, I'm going to give you a cheat sheet about the cytokine families. So we're going over this in a cursory way to make sure that everyone understands what cytokines and chemokines are. Yeah? What?
Are you holding a review session?
Review sessions. When would we have review sessions? Thursday I can probably help. No, not Thursday. Thursday I have my normal office hours after class, so you can come see me anytime between noon and 5 p.m. on Thursday night. I'm in my office because I got to go to a GB meeting on Thursday night. Friday I have office hours. I can hold office hours this week afternoon. I'll make sure that I post this so that you guys can know where I'll be. Monday, I'll probably be in my office for most of the day, so I'll make sure that I post when I'm in my office on Monday so that you're prepared and ready. But if you're going through your review sheets, the study review questions, those are what to look at right now. Do you know what I'm asking you? Can you answer those questions? If you can't, that's what to ask me on Thursday, or email me, or come see me to expound. Okay? Have a good one.